Welcome everyone to this webinar, and we're very pleased here to have many distinguished panelists, which I'll introduce in a moment. I head up the state and international government affairs function at Amgen, and I am based in Washington, D.C., and I'll start with Patrick Frey, who is Director and U.S. Lead for Global Regulatory and R&D Policy at Amgen. Prior to that, he worked at FDA's Center for Drug Evaluation and Research as Director of the Office of Program and Strategic Analysis. He was later Chief of Staff for the Office of New Drugs. He received his bachelor's in chemistry from University of Pittsburgh and a Master of Public Policy from University of Michigan. So he will kick us off. And I'll introduce the other three panelists now. John Quisel is the CEO of Disc Medicine, which is a biotech company that focuses on treating serious hematologic disorders. John has guided me through various stages, so it became a publicly traded company with a valuation in excess of $1 billion. Previous to that, he worked at Acceleron Pharma, helped build that company, and it was acquired by Merck for a very high price. And then he's also worked at law firms, Ropes and Gray, Foley Hoag, practicing intellectual property law and biotech. He received his degrees, undergraduate from Harvard, a master's from Stanford, PhD from MIT, and a law degree from Harvard. So I will continue on now with Elizabeth Wood. She's founder and CEO of Jura Bio, where she leads a team of scientists and engineers who create cell therapies for oncology and autoimmune disease. She's been there working for a long time, working with very complex technologies, and Jura's vision is to transform the way diseases are treated and cured with cell therapy. Elizabeth received her undergrad degree in physics from Harvard and a PhD in physics from the Technical University of Denmark. I will move on now to David Cheng, who is the Chief Technology Officer and Founding Director of Computation at Arbor, a gene editing company developing curative therapeutics for patients. Prior to Arbor, David worked at JP Morgan and Goldman Sachs, and he developed market-leading quantitative automated trading strategies. He also worked at Google at Parallel Time, and he was a member of the four-person team that represented the United States at the International Olympiad in Informatics. David received his undergrad in computer science from MIT and as well a master's in engineering. I'd like to welcome all of you panelists, and apart from the Amgen folks, everyone is an RSI alum from CEE. So we're just delighted to have you speaking to the group. So we'll turn it over to Patrick, who will give us like a 101 on drug development and working with the FDA and how we get through those processes. Panelists can then chime in with their experiences with the FDA, with their products, with their experience, and also talking about what they've seen changing along the ways and however you would like to add your so with that, Patrick, please proceed.
Well, happy to kick things off here. I'm going to talk about 3 different topics and breeze through these. basically, like a lot of what Kathy said, interacting with FDA during development, a bit about FDA's expedited programs. That's often an important topic. And then a little bit about how we got to where we are right now with FDA from a regulatory decision-making standpoint. First topic. So the graphic that you see on the right, that's pretty standard for what we typically see with respect to the drug development life cycle. And what is FDA, what's their role during drug development? Obviously, they review and approve or not approve products, but they have a pretty significant role to play during development. And you see there the phases that we often talk about during clinical development, phase 1, 2, and 3. During all those phases, FDA's responsibility is ensuring the safety and the rights of trial participants. But then when you get into the later phases like phase 2 and 3, FDA adds to its responsibility, ensuring that the quality of the scientific evaluation of the drug is going to be sufficient to assess the safety and effectiveness. I've got a few numbers there, 1 through 5, that I overlaid on top of the figure, and they track to the numbers on the left side of the slide, and they refer to the milestone meetings that typically occur between a sponsor and FDA during drug development. So the pre-IND meeting, that is where a sponsor is discussing plans with FDA for the initial investigational new drug application, or IND is the acronym. They're talking about safety signals that might be identified during preclinical work, what additional preclinical work is going to be necessary to maybe further investigate them, plans for developing the product, discussing the sponsor's intentions to pursue one or more of the FDA's expedited programs that we'll talk about in a bit. At the number 2, at the end of phase 1, that's an end of phase 1 meeting that can be held for certain products, where discussions are held regarding the planned phase 2 studies, the pharmacokinetics and pharmacodynamics of the drug, dose selection, the adequacy of supporting non-clinical data, and continuing discussion about any expedited programs that the company may want to take advantage of. At the next milestone meeting, the end of phase 2 meeting, you're discussing, as expected, what are the planned phase 3 studies that are intended to support registration of the product. This arguably, I think, is probably the most important milestone meeting during development. The company is also talking to FDA about inclusion-exclusion criteria of those studies, the endpoints to be used, any risk management that thinking that may need to occur at that time based on the accumulated non-clinical and clinical data up to that point during development. And if the company is planning to pursue accelerated approval as the approval pathway for the product, they're probably talking about confirmatory studies at that time. And we'll get into a little bit more detail about accelerated approval in a minute. Then basically after the clinical development phase has kind of run its course and come to a conclusion, the company is planning to submit the application, and FDA and the company sit down on a pre-submission meeting to discuss the format and content of that planned submission, statistical analysis of the clinical data, risk management actions that might be needed to facilitate FDA being able to reach a favorable benefit risk assessment for the product. And then the last time point actually occurs during FDA review for a certain subset of applications and products that FDA receives. We call them new molecular entity NDAs and original BLAs. So in general, FDA has never approved, the molecule has never been approved or marketed. And for that special category that FDA actually reports on every year, because it's always a focus, that's where the most innovative medicines are being approved by FDA. There are special time points of a mid-cycle communication and a late-cycle meeting where the company is meeting with FDA during review to talk about how the review of the application is going, what additional questions does FDA have, and would there be additional data that the company has that could be submitted and potentially reviewed during that first review cycle, because that's kind of the goal of, if the product can be approved, approving it in the first cycle of review is efficient for everybody. So in terms of how the whole meeting process works, a company requests the meeting, FDA decides whether to grant or deny the request. If they grant the meeting gets scheduled. Then the sponsor needs to submit a briefing document to FDA that's going to contain questions for FDA. FDA will review that material and then send back preliminary responses to the company of those questions. And then the company needs to review that information and decide whether the meeting should still be held or whether those responses are sufficient and the meeting can be canceled, which happens a fair bit. If the meeting is held, then the company gets meeting minutes from FDA. And something relatively new in the Human Drug Review Program is that if there are still outstanding issues that need clarification on the part of the company, there's a time-limited opportunity for additional follow-up with FDA, not to introduce new information, but only to clarify the discussion at the meeting if there are any doubts. And then the one final thing to note here is that for certain meetings, FDA has the ability to convert them to a written response only. If they feel that that's the most appropriate way to answer the company's questions and an actual face-to-face meeting isn't necessary, it's an important tool for FDA to use to manage their workload. And a company will get actually a faster answer than if they would hold a face-to-face meeting. And the written response serves as the final output of that meeting process for the company. So let's go to the next slide, and we'll talk about a few best practices and tips for engaging with FDA during development. This list actually came from a sit-down we had with project managers at Amgen's Thousand Oaks campus. It's a program that every year a certain number of project managers can go and travel to a company site and meet with representatives from the company to have a deeper understanding of the drug development process from the company's standpoint. And this is what we heard from the regulatory project managers, that they're your primary point of contact as a sponsor, your primary interface with FDA. That establishing the relationship with the RPM and agreeing on what that communication strategy should be between the RPM and the regulatory affairs professional at the company is very important. Giving your RPM a heads up when you're sending important submissions like a new meeting request or protocol submissions where you have actual specific questions for the review team. FDA also said that not everything needs a formal meeting with them, that it's appropriate to just contact the RPM, what are your short list of questions for the review team, and ask their view on what the most efficient way is for getting an answer from FDA. And then when submitting protocols and amendments, note the questions that you have in the cover letter. That facilitates the RPM in their triage within the review team, because there are 5 or 6 different disciplines that are part of that review team at FDA, and the central point of contact is that regulatory project manager. And then, like I said before, making sure you review FDA's preliminary responses as part of that meeting process and make that decision about whether the meeting is really needed or not. Talk a little bit about FDA's expedited programs. I'm going to focus on the 2 programs that I put in the black box, just in the interest of time. Breakthrough therapy, and I'll just kind of talk down the column for breakthrough therapy and then move on to accelerated approval. The criteria for breakthrough therapy, and your product needs to be intended to treat a serious disease, and you need to have preliminary clinical evidence that indicates that the product demonstrates substantial improvement over available therapies. Features of that program are intensive guidance on your drug development, a real organizational commitment to that product and developing it in the most efficient way with the company. FDA often talks about breakthrough therapy program as being an all hands on deck mentality, and they do have that kind of attitude internally. They will meet more frequently with the company during development to make sure that it's run efficiently. But if something changes with the product itself, if later phase data indicate that maybe it does not have that substantial improvement over available therapy, or maybe the available therapy has changed, that designation can be rescinded. A good time to submit this request to FDA for the designation is with your IND or after that. But once you get later into development and beyond that end of phase 2 meeting, then you lose the benefit of the breakthrough therapy designation. So it makes sense to request it as early as possible. And FDA does have, since they received this program back in 2012, 2013 by statute, they've added a component of the program where you can seek preliminary advice on whether or not FDA thinks it makes sense for the company to submit the designation request for breakthrough therapy. So you can get that preliminary advice and informs you whether or not it makes sense to submit that request to FDA. Moving on to accelerated approval, the criteria for that program also must treat a serious condition, and it generally also provides a meaningful advantage over available therapies. And the effect that you're demonstrating is on a surrogate endpoint that is reasonably likely to predict clinical benefit. The features of the program include review of the promotional materials before they're released, should the product be approved. And because the approval has some uncertainty compared to traditional approval, which is actually based on a clinical endpoint, the company needs to run confirmatory trials to verify and describe that clinical benefit. Clinical benefit defined as how a patient feels, functions, or survives. So that criteria is not met by a surrogate endpoint. But in your confirmatory trials, by running them on a clinical endpoint, if you're able to verify that, then you convert from accelerated approval to traditional approval. And if that confirmation doesn't happen, products that are approved under accelerated approval can be subject to expedited withdrawal, and that has happened in the recent past. Timing of this, like I mentioned before, these conversations need to occur early if a company is planning on using the accelerated approval pathway. And that conversation includes, understanding the surrogate endpoint that the company plans to use, its connection to clinical benefits so that FDA can determine that the reasonably likely threshold has been reached in their mind, and then also to discuss plans for the confirmatory studies that need to be conducted in the post-market setting. So this table, if you're interested in the gory details, I have the link at the bottom of the slide, and these slides will be distributed after the meeting because I slightly abridged it for this presentation. And we'll talk a little bit about how we got here. So from the perspective of drug regulation, it goes all the way back to 1906 with the Pure Food and Drug Act. In 1938, then we got the more modern law of the Food, Drug, and Cosmetic Act that has been amended since then, and that's FDA's authorizing statute. In the early 60s, the FDCA was amended to establish the effectiveness standard. And then there was a little bit of a quiet period for about 30 years until the drug lag that was occurring in the United States compared to other regions and other countries really needed to be addressed, because other countries, their patient populations were having earlier access than Americans were to drugs being developed. So that in 1992, Congress passed the Prescription Drug User Fee Act, which authorized FDA to collect and spend user fee dollars from industry to make the drug review process more efficient. As part of that law, it must be renewed every 5 years. So since 1992, we've been in the middle of 5-year cycles where the Food, Drug, and Cosmetic Act can be opened for possible changes. And in the next couple of years, we'll enter the cycle to figure out what the next 5 years of the program looks like. It's interesting to note that in the early years, a lot of the legislation came as the result of to address tragedies and safety problems. And it's kind of like, you think about it as a pendulum. When I was at FDA, that's kind of how we talked about it in terms of which way is the pendulum shifting from safety to innovation. If you look back to 2007, when FDA got the FDA Amendments Act, The focus of that statute was on safety because FDA was coming off of the Vioxx safety problem, the Avandia safety problem. So a lot of the new authorities that FDA got in 2008 were safety related. Then you flash forward 5 years and the whole conversation changed. And the name of that law is the FDA Safety and Innovation Act, where most of the language in that act pertains to innovation. And that's kind of where we've been ever since then, a real focus on innovative products, speed to market, etc. Because we start with the statute, I kind of want to walk you through how we get to FDA's regulatory decision-making framework, and everything starts with the statute. Both bullets actually come from the statute. The first one establishes the substantial evidence or the effectiveness standard that FDA uses to conclude that a product has been demonstrated as effective. And then the second bullet further defines what substantial evidence means. And like I said, both of those bullets are in the statute, but I've also bolded adequate and well-controlled investigations because that is not defined in statute. So Congress left that up to FDA to further define, and we can go to the next slide, which agencies often do. They write regulations to further implement the law, to provide an additional layer of detail about the law. And FDA did that in writing regulations about what is an adequate and well-controlled study. And you can find that in Title 21 of the Code of Federal Regulations. I did not cut and paste that in here because it's quite a long regulation. You can peruse that on your own. But these regulations interpret the law at a more detailed level, and they're subject to notice and comment. So FDA needs to go out with a proposed rule or a proposed regulation, and then collects public comment on it before then it finalizes that regulation. So that gap is basically that I talked about is basically, uncertainty. You can think about it that way as well. And despite, what science and medicine contributes, there are residual uncertainties that FDA has to consider. And then this leads to the introduction of, an individual's judgment, how their values might weigh into the decision. And individuals can weigh the same information very differently based on their own judgment and values, and often lead them to different conclusions. FDA considers other factors in its regulatory decisions to help manage this, like disease severity and its assessment of existing treatment options. I saw this myself when I was at FDA. It was a product, it was about 10 years ago, I think, and it was for diabetes. And there are a lot of treatments out there at that time for diabetes. But it had an animal signal. There's a safety concern from animal data. And most of the folks on the review team felt that, based on the totality of information and the benefit that the product provided, that there was a place for this in the treatment armamentarium for the product. But one individual, looked at the treatment armamentarium and thought that, she wasn't sure that there was a home for this product in the armamentarium because of that animal signal. So that had to be discussed and debated in the review team to resolve that issue. But again, faced with the same information, but just viewed it differently based on an assessment of the treatment armamentarium in that case. And I'll add too that patient perspectives can help inform and resolve some of this uncertainty for FDA. In CEDAR and CBER, they've had a program for a number of years now called Patient-Focused Drug Development. That's the link that I've provided there. And that can help resolve the uncertainty. And I should add too that the input from healthcare providers and experts in the field that FDA can get through advisory committees, that can also help fill that gap in understanding from those experts how this product will be used in practice. And you put everything together, and this is a project I was involved in when I was at FDA in developing the benefit risk framework. And you might say, well, okay, what's a benefit risk assessment? You've talked about a lot of other things, but this benefit risk assessment, what is it? And going back to what I said about substantial evidence and how that's defined in statute and then further amplified in regulation, that when you talk about FDA approving safe and effective drugs, effectiveness, as I've said, is well defined in statute and regulations and then in guidance that FDA has put out. Safe is really nowhere defined. So FDA has long, interpreted that its conclusion about whether or not a drug is safe is that the drug has a favorable benefit risk assessment. And the table that you see here on the left, and then the guidance that was published not all that long ago, is what reviewers are filling out and completing and inputting information here about the product that they have under review. If you look at the dimensions, you can walk through the different dimensions and basically turn them into questions. The reviewers need to ask themselves, how bad is this disease? What's my assessment of the current treatment options for this particular disease? The product under review, what good does it do? And then what are the things that it does that aren't so good? And can I manage them or mitigate them in any way through risk management? And all this information then gets integrated, and they write a benefit risk integrated assessment that you can find on drugs at FDA where review documents are published. So that's it in a nutshell. The final slide I have is links to some resources about the content I've discussed. And I think I'll stop there and look forward to your questions later on.
Thank you, Patrick. I know there was a lot to unpack there, and you did it in a very efficient way, setting the stage for what is the process at FDA, what's the framework they use. And now I'd like to turn it to our panelists and hear from them individually in the order that I introduced them. I think John, Elizabeth, David, what are your experiences with the FDA and with your in your companies, either past or present, that you would like to share with our audience? And you can certainly take it in any direction that you see beneficial.
Sure, thanks. I'll pick it up. So thanks for that introduction, Patrick. That was a fantastic review, and there's so many different threads that could be pulled on. So I've spent about 2 decades as, on the company side as a consumer of these interactions with the FDA. And I guess during that time, you would characterize it as the innovation-friendly side of the FDA. And I think it's overall been a successful regulatory regime for balancing the interests of the companies and patients and, guarding the safety of the American public. I think it's overall done well. There's always controversies that you can point to. But I guess I'd pull on the thread of, lurking behind that process of FDA interactions. It's astonishing how rigorous that is, how humbling the clinical trial process is, that these great scientific ideas, come up in the laboratory, become converted into a product. Huge amount of rigor that goes into turning those products into something that can be just made in a reproducible way again and again. And there's a huge amount of effort and money that goes into that. And then you run these clinical trials, and the rigor of those trials just far surpasses the rigor that goes into any of these mouse experiments or whatnot. And again, people are surprised by the results they get. And sadly, more often than not, the results are negative. But every once in a while, things are incredibly positive. And so, the group, the different kinds of people that have to get involved at each step along the way, in terms of the scientists who discover the molecules, the essentially chemical engineers who have to manufacture the molecules, the toxicologists who sort out the likely safety before the clinic starts, the clinicians and statisticians who design the trials, figure out the appropriate endpoints, the way to measure efficacy in the human setting, and then the people who mediate the interactions with the FDA. You have to bring all these people together at different stages, and literally costs hundreds of millions to billions of dollars to do this for just one product. So it's an amazingly intense process that we go through that just draws in so many different kinds of expertise. So I just thought I'd highlight that. I guess one other thing I'll share is that it really is a life and death process that the FDA goes through. You talked about expedited pathways, and the pretty common fact pattern that the company and the FDA faces in a good situation is you have a drug and it's worked really well in that phase 2 study, right? It's often the first study in the actual patient population. You see this very strong signal of efficacy, and safety looks all right. Maybe it's 100 patients. This is a specific example I lived through, actually. And the efficacy is better than has been seen before. And if you're in oncology, there's been a big push there. Often you see the FDA grant approval on that kind of package and allow that drug to reach patients broadly. In my instance, it was a non-oncology indication where mortality was set at about 5 to 7 years, so just as deadly as a cancer, but sitting outside that kind of special zone that oncology has enjoyed. And the FDA's decision was not to approve the drug on that initial study, and so a phase 3 trial was run. It took several years, and it was about 300 patients, and remarkably and sadly, there was about a mortality difference of about 30 people between the placebo group and the treatment arm in that phase 3 trial. So you literally had 30 people whose lives who didn't receive therapy in the name of science, basically to get a sufficiently rigorous answer about this balance of efficacy and safety. And that just illustrates just how important what the FDA and how tricky it is to decide, right? If they approve that drug and it turns out that they're wrong and the efficacy is not good and the safety is not good, you then have patients who suffer for that in the marketplace. But on the other hand, when they withhold that approval and you have to do another study and then you prove with mortality that the drug is actually working, there's regret that it wasn't approved and allowed out to the patient population sooner. So it's a remarkably high stakes business that we're all engaged in here.
I'm happy to pick up on that, John. And I know the saying, and probably everyone else does, but in these optimal stopping problems of when to approve a therapy or not, the FDA can only sort of be too early or too late. It's just impossible for it to get that timing exactly right, except I guess with the full insight of knowledge, and in hindsight. I get to talk today about an exciting class of medicines, exciting for patients, I think challenging, but excitingly so to the FDA, which is the regime of personalized medicine. The personalized medicines I pursue all within cell and gene therapies. These are therapies that have, only been made possible in the past couple of years, thanks to, tool building companies like David's, where we're able to go in, change genomes, edit cells that we introduce to patients. But, in those pivotal studies that John was talking about, you might have 300 patients, 3,000 patients in a phase 3 study. What about the regime where you have maybe 3,000 patients total for your drug, or in fact, it's only a single patient that is ever going to receive the exact formulation of the drug you're making because it's specific to a neoantigen that's only ever been seen on their cancer tumor. And this is sort of a cancer vaccine setting. And that's where, I think about and where I work is in the world of antigens, which are extremely specific to people. So here you are, you're a person, you're about to receive a drug, and you're going to be the phase 1, phase 2, and phase 3 trial, in effect, of the drug that has been made just for you. And so this is obviously a different regulatory regime, and the FDA's answer to that, Peter Marks and others, has been really forward-looking. I think that the people in gene therapies, rare diseases, especially among children, have been really pushing forward our ability to say, okay, how do we address these, much needed, fast-moving therapeutic modalities? CBER has risen to the challenge, I think, of trying to address this, but fundamentally, it's a sort of unsolvable problem. That we can only hope to do our best on, as the FDA always only hopes to do their best on. So in this regime where, you sequence someone's tumor, you have one shot, a few weeks to come up with a few epitopes, to choose those epitopes, to put them into the patient, and that patient will act as their own control. Then you're in a regime where, adaptive clinical trial design can help a little bit. An emphasis on what's usually considered preclinical regulatory studies can help a little bit, because what you really have to nail in a modular therapy like that is the process by which you characterize the patient's target, you characterize the therapy that you make, you rationalize and normalize the whatever efficacy readouts you can get ahead of time before you go into that patient. But sort of like once you've set your recipe, it's that over which you get approval, as opposed to seeking FDA approval each time you need to go after a new antigen in a different patient. I mean, I will reiterate that for us as patients, for us as loved ones with patients, with patients in our families, it's really exciting to know that, diseases where there have been like treatments of no last resort all of a sudden might have this class of personalized therapies coming to answer the challenge of metastatic cancers or gene driven diseases. And so I think it's a challenge that we're all actively engaged in solving, and that, as a founder and sort of scientist working in the space, to me, all I can hope to do is build the tools by which I, predict and perform the making and characterization of my therapies as best as possible. And then, hope that the FDA is there to walk hand in hand with me as we deliver that to patients. I'll pass it over to you, David, so you can talk about the challenges of even making those tools that allow that to happen.
Great. Thanks, Liz and John. And yeah, maybe I'll start off with a bit of a tangent. I think John made a point that it takes a village of diverse experiences to develop a drug. So I'll just start off by talking a little bit about myself, some background. You mentioned Goldman Sachs and JP Morgan. So what's that all about? So I went to RSI in 1999 and then studied computer science. And my first job was in finance almost 20 years ago. And what attracted me about to finance then was being on the ground floor of the electronification of a particular market. And back then, this was the currencies market. And that really allowed for rapid innovation. It meant that every few years, the industry would really have to reinvent itself. And just a couple quick vignettes of that is that I saw the amount of data increase by orders of magnitude multiple times. For example, the amount of prices coming through the wire went from 1 price per second to thousands of prices per second. Or the pricing granularity changed from hundredths of a cent to thousands of a cent. So each one of these changes was really an opportunity, because that meant we had to throw away the existing trading signals and strategies and really hunker down and understand what's going on, establish new A/B tests, and whoever could get on top of that first would really become the leading player in the market. So I share that bit of a background or kind of a tangent because I'm really excited about drug development today. And I see a lot of changing trends. And I think it's just a great opportunity to be in today. So just a couple examples of that are just a few years ago, Google DeepMind developed AlphaFold and made it available. And it's really a game changer. It allows folks to get actionable insights from the structure of a protein. And what would previously take potentially months of work and hundreds of thousands of dollars can now be done in just a few hours with just a few dollars of cloud computing resources. Or another example is that DNA sequencing costs, they've gotten really cheap. And these days, it's less than 0.01 cents per million base pairs. And the field is continuing to innovate. There's new technologies like the nanopore. And that's really important because it allows high throughput screening methods with digital sequencing-based readouts. And that can allow us to get to the scale of data for complex molecules like enzymes or DNA or RNA to apply AI techniques to really learn the rules and maybe get a deeper understanding of all the many factors that can play into the molecular mechanisms of actions. Using those insights, then we can engineer and design, or I guess what's the latest buzzword is to generate, more functional molecules. So that's definitely a hope of where the field could be going. But so far, it's just been talking about the capabilities or activities. Really, I think what was great about Patrick leading off is we really have to be goal-driven in this industry. When we're building a bridge, we have to know where we're landing, and we really have to make sure that we've really pinned down what is the disease, why are we going after it, what is the target, and how can we work backwards, from that particular goal. And maybe I'll walk just through some perspectives that, of course, colored by Arbor, which I've helped build over the last 6 years, which is a gene editing company. It's founded by another RSI alum, Feng Zhang, and we're in the gene editing space, which is quite new, similar to what Liz was describing. Gene editing together with gene therapy and cell therapy makes up what's sometimes called the third pillar of medicine, after small molecules and antibodies. I think what's exciting about gene editing is that with it, we have the potential to really change the paradigm of medicine. So instead of treating the symptoms of a disease, for example, having a patient take a pill a day or get an injection every month, we can allow these so-called one and done curative medicines to halt or in some cases reverse the progression of a disease with a single administration. We can make that more concrete. At Arbor, one of our areas of focus is tackling diseases in the brain. For example, neurodegenerative diseases. First of all, they're devastating, but secondly, many of them have textbook genetic causes. And if you look at the history of drug development in the area of the brain or neurodegenerative diseases, small molecules and antibodies have just had a really tough time really showing that safety and efficacy that we need. So we're really excited that gene editing can give us the tools to really move a needle in this space. So once we've kind of articulated a space, kind of honed in on some diseases, then how do we walk through the preclinical process of developing a genetic medicine? Maybe I'll kind of gloss over the whole topic of indication selection, and that could be a whole hour topic itself on how folks kind of go through the disease epidemiology and think through the age of onset and age of detection and the tractability of a disease. But really, from a drug developing, especially a small biotech perspective, we have to focus. And as part of that, we have to think through what are the different risks that we're willing to take on. And there's broadly speaking, for genetic medicines, 3 big buckets. One bucket is target risk. We want to make sure that if we're intervening at the genetic level, we're really hitting a genetic driver of a disease. We're not just touching something that's correlated with people who have the disease. So we want to make sure that the target risk is pretty understood and solid. Another bucket of risk is delivery. How are we delivering the genetic payload, the gene editing machinery, whether it's as an mRNA or in some other nucleic acid form? What is the delivery vehicle? What is its biodistribution? What is the safety of the delivery vehicle? Those are some considerations. What are the organs that it can reach? And thirdly, of course, is what is the edit that we're trying to make? How easy or how complex is that edit, and how much efficacy are we able to achieve with that edit? So that really drives how we think about defining a target effector profile. What is the edit we're trying to make? And usually the first thing we do is start out in an easy to work with cell line, like a hex cell. And we work with many different types of gene editors, and we can do some large scale screens to identify, well, how close are we to the target edit that we're trying to make. After we can engineer and optimize our molecules in that easy to work with cell line, we then advance into typically primary human cells, and from then we go on to in vivo studies. And that really gets into working backwards from, well, what is the submission that we will ultimately make to the regulatory authority, to file an IND. For gene editing, there's a precedent of other companies that have been through this process, and that breaks down, the package into three big areas. One of them is genotoxicity. We need to characterize off-target editing in human cells to make sure that these editors are only making the intended edit, and there's no concern about making edits elsewhere in the genome. A second bucket is efficacy. So we have to show that these editors are effective at treating the disease in an animal model, usually a mouse. And the third bucket is on the safety side, we have to run a tox study, typically in non-human primates, to understand the safety profile and determine any appropriate dosing for that. I think the last step before an IND submission is really to work out the CMC side of things, really to work through the manufacturability of the drug product at GMP grade, making sure that all the input reagents are well tracked, the quality of the purity of the material is good. I won't go too much into that, but as you can imagine, it's a whole thing, and it's a big effort in both time and expense. And maybe just some final thoughts before we shift to the Q&A is that I think it's still very much early days when it comes to developing genetic medicines. I think there are lots of orthogonal opportunities to move the needle. Some of them could be, even on the business side, finding ways to make these more affordable, or figuring out ways to change the payer business model. Because with the one and done treatment, then that actually creates a wrinkle in how a lot of our existing structures are set up. I think on the scientific side, being able to discover and validate new genetic drivers of disease is also a pretty exciting area. There's just a massive amount of new genotype, phenotype data being collected. About 15 years ago, it was a big deal when the Broad Institute collected 1,000 genomes. And then about 5 years ago, the UK Biobank became available with about 500,000 genomes. And right now there's ongoing projects that are on the scale of sequencing millions of people with their paired medical histories. And by crunching the associations of genetic variations to diseases, I think that we'll find is that more and more diseases, or at least subtypes of diseases, will really be genetically driven. And they may be driven by mutation in one gene, which would be called a monogenic disease. We may find that some diseases are more complicated than that. Maybe they're polygenic. And we also find that there will be a lot of spontaneous diseases, maybe affecting cells at the RNA level. But either way, I think these discoveries will represent really new and exciting hooks for drug development efforts to be able to tackle these diseases. So maybe I'll just, end there on kind of a hopeful note and turn it over to the panel for open questions.
Sorry, I did come in a little bit late, so I apologize if I missed this. I heard at the beginning discussion about the accelerated pathway for drugs, and that is, I think, often very essential and exciting. There is some issues with the follow-up sometimes, I gather. I'm just a run-of-the-mill rheumatologist, so I don't know a lot of these details. But I guess when you have that, and especially I guess when you have these very low-end initial approaches to patients, what kinds of things should change, need to change, are changing to balance that pendulum somewhere in the middle, to make sure you're getting the signals that you need, getting the information about the signals out there, because doctors aren't great about knowing science a lot of the time. So yeah, is there discussion about how to manage that going forward?
From what I've seen, it has been extremely important to patients, and it is constantly under fire. It feels like every year there's more attacks sort of launched against it. There is an obligation by companies that have received accelerated approval. I think Patrick mentioned this, to come up with a study after the fact, once it's in the population, to demonstrate efficacy and its ongoing safety. And the problem, as I understand it, has been that companies have been falling back, behind on that commitment to actually have the follow through in those studies and in sharing the results. Am I right about that?
Yeah, I think that's exactly the controversy. I guess I think that pathway has been the most robust in the oncology space, as I mentioned earlier. And I think there's been a lot of drugs getting approved on what are sometimes not placebo-controlled trials, where you're just looking at patients who receive drug, and you can measure what are objective responses in terms of tumor size and things like that, and so that led to a lot of enthusiasm for early approval, which does have, and Liz, you're kind of addressing, I think, hinting at that huge effect on investment in the biotech space, right? Because if you can cut the 3-4 years of a pivotal trial, right, these often take a long time in phase 3, it improves and see the potential to sell drug 3-4 years earlier, the financial return for an investor becomes dramatically higher. So there's this very powerful effect of the hope of accelerated approval in bringing capital and investment into the development of new drugs. So it has this feedback loop that's potentially very powerful pro-innovative effect. And then, the system has suffered sometimes where those confirmatory trials, like you said, Liz, sometimes the companies just, the incentives may not be there to really go and run that trial expeditiously because you're selling the drug, and you maybe don't even want to know the answer to that confirmatory trial. But in some cases where that trial has been completed, the answer is the drug didn't work. And that's not great for anyone either, because suddenly now you have some patients who are taking the drug and maybe feel it is working, right? And they don't want to have it pulled out of their hands. The FDA doesn't, I think it's very difficult for them to then say, okay, well, this drug we now know scientifically doesn't work, and we're going to withdraw it from the market. That's a mess. So I think there's been a push now actually to increase the rigor of the phase 2 trial that would be needed to support an accelerated approval. And I've certainly seen that in some of the programs I'm working on, that it feels like the bar is higher. And, I think there's benefits to that as well.
Yeah, I think I'd just add, accelerated approval, that program is a really important tool, for drug development for patients. And the past 10 years or so, I think you could say it's been predominantly used by the oncology group at FDA. We published a paper last summer in the DIA journal, and we took as much public information as we could to kind of like piece together a story of the first 10 years, the second 10 years, and the third 10 years of the program. And encourage you to find that, or we can send that as a follow-up. But because this program has been used predominantly by the oncology group, they've taken the initiative to make sure that the program is being managed well, because both companies and FDA have a responsibility to the program. And that's why you might, if you follow the trade press, you've seen FDA talk about dangling approvals and delinquent approvals and stuff like that. And when they are faced with results from the confirmatory study that make them scratch their heads, they're taking more of these drugs back to an advisory committee to have experts weigh in on this. And that's been a really important tool, to understand what the view is of experts. And then if FDA, the one thing FDA said is that a failed confirmatory study doesn't necessarily mean a failed drug. There might be another path forward. Maybe there was, errors in the design of the original confirmatory study. So there's that conversation that will still happen between FDA and the company. But if FDA concludes that there's not a path forward for the drug, then their next step is to request that either that indication, if that drug has multiple indications, or the drug, if that's the only indication, be removed from the market. And then if the company has the ability to challenge that, there have been recent legislative changes in the program that have made an attempt. We're going to see how this plays out, to allow for expedited withdrawal and from the market. And FDA also now has the tool to require that confirmatory studies be underway at the time that it grants the accelerated approval. So there's a couple of tweaks that have been made to the program, and we're going to have to see how this plays out over the next couple of years. But a really important tool, and both sides kind of have a stake in seeing it preserved.
Thank you. And actually, we're at the end of our time here. I'm sure we could take more questions if you get them. I'm happy to have the panelists look at them and answer anything that we can. But thank you, panelists and participants, for joining us today.